All right. Good afternoon, everyone. Thank you for the kind introduction. I'm Josh Wood. I work with Professors Eric Popp and Joe Lighting, and today I'm going to talk to you about our work using graphene as atomic shrink wrap for nanosandwiching biomolecules. And so I'm going to go over briefly why graphene is a unique two-dimensional membrane and some of the work we've been doing with nanosandwiching water and other biomaterials with a unique transfer method that we've developed in our lab. And then I'll conclude with some remarks about how we can study protein hydration and denaturation between these graphene sheets. So what is graphene? Well, a lot of you might already know what graphene is, but just briefly, graphene is the mother of all carbon nanomaterials. It is basically how you make buckyballs, carbon nanotubes, graphite, depending on how you stack it, how you wrap it. And the way that graphene was originally isolated was called by the so-called Scotch tape method. It's very sophisticated. Use your tape and graphite and you get essentially a Nobel Prize. The isolation of it got these two gentlemen the Nobel Prize in 2010 in physics. But what's interesting about it is its electronic band structure. There's high mobility, high Fermi velocity. The bands are symmetric about the conduction and valence band. There's been a lot of work on this. And so to show you that band structure again, essentially if you look at it in the plane, it's linear around the what's so-called Dirac point, but it doesn't have a band gap. So while it's great for certain applications like radio frequency transistors, or perhaps in heterostructures like graphene integrated with boron nitride or molybdenum disulfide, it's really not great for conventional silicon-based CMOS electronics. What it is good for is flexible touchscreens. This report from Samsung showed that graphene can be used in a 30-inch touchscreen application. But what we're more interested in is the fact that graphene is this perfect membrane. It's not stretchable in plane, but it's very deformable out of plane. And here is a recent report where we've been able to suspend graphene over a trench in silicon dioxide. And so it's very good as a membrane, maybe not so great for electronics. So we're going to focus today on this particular application. So these membranes are pretty remarkable. They can entrap gases, liquids, solids, nanoparticles, the kitchen sink. Anything you want can be trapped under graphene. And there's been several reports looking at what you can trap with graphene. In this case, this report in Science was nanosandwiching liquid platinum between two graphene sheets. If you carefully make a graphene laminate structure, you can basically trap all gases and permeate water through it. And then these gentlemen actually use that to distill alcohol for some reason. You can use it to veil graphene nanoparticles, and this is another report from this same group here. But more interestingly is, well, if you put it over water, that it actually drapes perfectly conformally to water. And this is a recent report from our group where we discovered that it conforms over 1 to 2 molecular layers of water. Why this is interesting is that if we have water and we have biomolecules which love to be hydrated, it might be possible to study these biomolecules in more innate environments, such as those that they would exist in a cell, but down to the atomic level. So scanning atomic microscopy, atomic level information of biomolecules has been pretty hotly studied for 20 years. And essentially, no one's really gotten very good at it. The initial reports came in the early 90s where they said, oh, we have DNA on graphite. When actually, this is actually a feature of the graphite, not DNA at all. So several reports came out later on refuting these results. Electronic data for these molecules is very, very suspicious. Sometimes you get these bands which appear in electronics, sometimes you don't. So it's pretty inhomogeneous exactly what the results are saying. The interaction between water and graphene, on the other hand, is very complicated. So we might actually be able to investigate some atomic level phenomena between water, graphene, and biomolecules, which is very, very interesting. This recent report suggested that the graphene is transparent to its substrate in terms of its wetting characteristics. And finally, this graphene sheet, while it can deform out of the plane, if you put a biomolecule under it, it's going to exert a pretty substantial force on that biomolecule. And this has been studied previously for trying to look at secondary structure in proteins. So we might be able to look at some very unique phenomena at pressures that you cannot achieve in the laboratory using a simple graphene sheet as the means by which to do that. So how do you make it? Right? That's the obvious question. We want to make graphene. We want to make it on a large scale. And in our lab, we've developed this process using chemical vapor deposition on copper. We can get a large sheet up to several inches. We spin on a polymer. The typical polymer is polymethylmethacrylate. We destroy the copper. We clean off the residue. We get to a chip. We do a good job. We can pattern the Illinois logo. And we, once again, trap water, as I showed before. But more importantly is this polymer doesn't come off very well. It contaminates the surface. This has been studied several times, where getting rid of it using heating and annealing and using dissolution and solvents has been very challenging. So what can we do differently? Well, if you do the heating, you do a good job. You can get rid of most of the residue. You can see here. The problem is that this 400 degrees Celsius anneal is going to destroy pretty much any biomolecule you would want to encapsulate. Also, you find regions that are inhomogeneous, like here in this additional atomic force microscopy image. If you don't do the annealing, it's bad, like I showed before. You can see lots of residue here. And if you're looking at a 2 nanometer protein, you can't see it amongst this mess. If we use this polybisphenol, a carbonate transfer method that we developed in our lab, we can notice that the transfer is extremely clean. We notice no residues on the surface after a room temperature process. So the key takeaway is that no high temperature process is required. We can basically use this to enable biomaterial encapsulation. So what can we trap between these two graphene layers? We can make a huge sheet of graphene. We can transfer it cleanly. What can we trap? And the first order experiment is to see what happens when you put water between two graphene layers. And here we have a scanning tunneling microscopy atomic scale image of the graphene layer. And it's actually, they should interact. There's two graphene layers, and they should interact in giving a stronger long-range pattern known as a moire pattern. We don't see that, which suggests that there's water between the two layers. Also, over here, you can see it's very unstable. And this instability is kind of like the STM tip pushing on a waterbed. The water is mechanically pushing back against you, giving you a mechanical instability. We confirmed that it is indeed water from Fourier transform infrared spectroscopy. We can see here with isotopically labeled water that we've trapped water. And we've used Raman spectroscopy to confirm that there's no electronic interaction, that the two layers are decoupled. So the water is stuck there. What about if you give it really extreme conditions? You know, heating it up should drive off the water. Well, we went to 650 degrees Celsius, which is extremely high. And we noticed that when we did our measurements that we blew a hole in the graphene layer, but we didn't destroy the water. The water stuck there. So it and graphene are intrinsically linked. You're stuck in this unforgiving relationship forever, which we can then use to study some interesting hydration phenomena with biomolecules between graphene sheets. So what are the molecules that we look at? Well, we look at the tobacco mosaic virus because it's this rod-shaped virus, which is very easy to see under most microscopy standards. We also look at a couple of proteins, the NeutrAvidin and the methyl-binded MECP1 protein, and also DNA. This is those original reports where we're all doing DNA. So we're interested in looking at all these different systems. So we put down our biomolecules on graphene, in this case the tobacco mosaic virus. Its height's a little lower than what we expect at 10 nanometers. We put a graphene layer on, it drops about threefold in height. And this threefold drop in height is due to the pressure induced by the graphene layer. It's an immense amount of hydrostatic pressure that causes the biomolecule to be squished by the graphene. And so with that immense pressure, we can study proteins underneath extrema that can't be reproduced really reliably in the lab, and also learn some atomic level information about what happens to these proteins in these conditions. And so to that end, we're going to talk about protein hydration and denaturation between graphene sheets. So how do we put down our proteins? Well, we transfer a layer of graphene first. We put down a protein, in this case the MECP1 protein by methyl-binded domain, methylated to DNA. Its height's 5 nanometers, as expected. We also look at this NeutrAvidin protein, which is also about 5 nanometers in height. And then we put a graphene layer on. Something really bizarre happens. When we put the graphene layer, we notice that there are these craters, these depressions that are all over in the image. And what are these? Well, there's a protein essentially for every single one of these depressions. And that is due to the fact that the protein is having a long-range influence on the local water structure around it. Exactly why it's having that is the subject of our future simulations, which we put that same MECP1 protein, MBD protein, between two graphene sheets. The water is not shown here, did molecular dynamics. And when we did that, the protein essentially denatures within a few time steps. And the motion you're seeing here is actually the water interactions with the protein. When we elevate it to 200 Celsius, the same model, it denatures immediately, and it's pretty mobile due to the fact that the water molecules are fairly mobile at 200 Celsius. So this encapsulation essentially causes the protein to immediately denature. To to do a control against that, we look at the trajectories of the bonds, in this case for both the protein sitting on one graphene sheet and the protein between two graphene sheets. And we notice that at room temperature, it doesn't denature when it's on one graphene sheet, and at 200 Celsius, it's fully denatured, which is not terribly surprising. The melting point of these proteins is about 50 C. When we put it between two graphene layers in the nanosandwich case, it's partially denatured. The 10 angstrom threshold is the point where the protein unravels itself. And when we heat to 200 Celsius, it immediately is fully denatured. So this is suggesting that this configuration is implying a lot of hydrostatic pressure on the protein, which causes it to change its folding characteristics. So we looked at atomic force microscopy imaging to see exactly what happens when we heat these things up. And this is the protein in question at room temperature. Go to 45 Celsius, we can see the protein hasn't changed its shape, nor the water crater surrounding it. And at 50 Celsius, right when these things are supposed to be denaturing, nothing's changing, which suggests that these things are already denatured when they come down, and that the water moving away is actually just a hydrophobic interaction with the hydrophobic core of the protein. So to explain that, we essentially come up with a crude model wherein we have the hydrostatic interactions between two graphene sheets and the protein. They give us pi cloud. The hydrophobic core interacts with it. It's also under 1 gigapascal of pressure. When you put water there, the protein core denatures and folds the protein exterior. The water screens away, changes the water configuration, and ultimately we have less water around the protein's extrema. So to that end, we have accomplished a clean transfer method using polybisphenol A carbonate. We use that to entrap water, other nanomaterials, biomaterials, other applications. We looked at specifically two different biomolecules, tobacco mosaic virus and MBD. They get extreme hydrostatic pressures cause these things to change their conformality and denature underneath the graphene sheets. This affects the local regions of water in the vicinity. And so with that, there's a lot of people that make this happen, and I'd like to thank all of them and all of you for your attention. Thank you very much.
Well, thank you. We have time for a couple of questions, perhaps. Are there any questions from the audience? Well, I have a question.
Yes.
Normally, if you people make these sheets, you see these different regions, these cracks running over, and those are the regions where you have, instead of one layer, you have additional layers of graphene on top.
Mm-hmm. So the morphology is pretty dependent exactly how much water is trapped between it. What happens is the second layer will actually introduce there's a wrinkle in the second layer, and the water in that vicinity will agglomerate at the wrinkle. So a lot of those linear features you're seeing are wrinkles in the top layer, because these are mechanically transferred one on top of the other. So you accumulate a lot of water in spots where you've essentially mechanically wrinkled it.
Okay. Thank you. No questions from the audience? Well, let's thank Josh once more.
Thank you.